Well, good morning, grade 12s. Welcome to our new reality for a little while. I'm here going to give you a video lesson with nobody in the classroom, which is totally weird and bizarre, but there you have it. And since YouTube could only allow me to put on a certain number of minutes, I better get cracking. So guys, I'm going to be covering baby development as we start from conception, fertilization, when sperm meets egg, up to kind of the first month or so of development, and especially dealing with the first week or so. So I hope you've got your slides, your PowerPoint. You can fill in as you go, but I've got to go pretty quick. So you might just want to listen and then, or pause the video when you need to fill in your notes. And it'd be a good idea just to do that, so that keeps you focused and it helps your brain remember what you're hearing. So here we go. Let's drag this little marker over here. All right. First of all, this is a very interesting picture. This picture was taken when a doctor needed to do some treatment to a baby in utero, which they can do. So in opening up the uterus there, this little hand reaches out, and this picture went viral at the time because that's pretty cool. Instinctively, the baby's hand took a grasp of the doctor's finger there. All right, let me just see if I can play with the light a little bit, make that a little easier to see perhaps. Not sure if that helps or not, but here we go. All right, so let's remember the great sperm race. We had our sperm starting in the epididymis, and it travels through the vas deferens to the urethra, then enters that battlefield of the vagina, moving through the cervix, finding the uterus, traveling down the fallopian tube, which we also call the oviduct, and then hopefully it finds the egg there. And of course that would lead to what we call fertilization. So it's important to know for some of these process words where they occur. So fertilization, of course, is occurring in the upper reaches of the oviduct or fallopian tube, like right about there. Remember, guys, we talked about the head of the sperm, which contains the acrosome, and the acrosome contains those enzymes that enable the sperm to enter the egg, and it's only one sperm, so there isn't excess DNA that enters. In terms of our chromosome count, remember the sperm is carrying 23 chromosomes. The egg has 23 chromosomes. So when we get that first single cell, which we're going to call the zygote, we've got to figure out a way to stand so you guys can see so it's going to take me a little while to play with lighting so that you can see the board properly. It's more important for you to see the board than for you to see me, in a sense, right now. So I'll try to stand where the board gets lit up properly for you. Maybe actually if I press, so the camera is making sure it's focusing on the board. Okay. So quick overview, guys. It's going to take about a week for the first single cell called the zygote to travel down the fallopian tube and make its way nicely to that pillowy endometrium for it to carry out its further development. All right, so the zygote, which has 46 chromosomes, we use this term diploid, guys, and we're going to hear and see more of that term in the next chapter when we talk about cell division. But the zygote is moved down the fallopian tube with the help of cilia and some of those peristaltic contractions that we learned about with the digestive system. It's going to take, like I said, about a week or 6 to 8 days to move down the fallopian tube, and while it's doing that, it's going through these things we call cleavage events, which are essentially cell divisions. So the zygote, which is one cell, will divide very quickly into 2 identical cells, which will then become 4, which will then become 8, 16, 32, you get the picture. And what's interesting is the actual cell doesn't get any bigger as a whole when it's just numerous smaller cells that it's dividing. But the zygote, or The mass itself doesn't get any bigger. And all the cells at this stage, guys, are identical. At this stage, as you can see by the bottom line here, if some of those cells are able to break free, since they're all identical, they all have the potential to become a clone of each other. And so, of course, that's how you're going to ultimately get identical twins. Now, that's an important term, guys, that as the zygote divides and you get to about the 32 cell stage, we actually give a different name to the zygote. Now it becomes what we call a morula. Easy way to remember is morula has more cells. By day 7, we, after more division and some specialization of cells, which we'll get to in a minute, the morula becomes what we call the blastocyst. And when it's a blastocyst, we're now at the uterus. And the blastocyst, so we would be right here, there's that morula at about day 4, cell mass size about 16 to 32 cells. When entering the uterus, it becomes the blastocyst, and that's about day 7, so about 1 week. And that's when implantation is going to occur. And a good little memory trick is the blastocyst needs to almost like blast its way into the endometrium. It makes a nice little pit for itself, it attaches itself, and then it's going to continue its development. And so that is implantation. So let's take a look at this blastocyst itself. Here's the blastocyst. You'll notice that we have these green cells. So these green cells are actually the embryonic cells. Remember we said each one is identical. Each one could become a very clone of each other. And we could get those identical twins. We now have some different cells called the trophoblast cells around the outside, the purple cells here. And they're going to become important. They will become cells that we call the extra embryonic structures that are going to be there to support the growing embryo as it develops. Over here you can see that some of these cells are going to become what we call the chorion, the amnion, and the yolk sac. And it's going to take about a month for these things to more fully develop. And we're going to talk to about each one of those in just a minute. So here again, we have the endometrium lining, and we have the blastocysts. We have those embryonic cells called the inner cell mass, and then we have this cavity surrounded by the trophoblast cells. I'm going to take this moment, guys, to plug one of the videos I have in the Google Classroom on embryonic development. It's really good at going over this development for the blastocysts and those extra embryonic membranes and exactly what they are going to do. So I really encourage you to check out those videos. All right, so here we have a quick recap of what we talked about so far. We have ovulation when the egg leaves the ovary, enters the oviduct, fallopian tube, meets the sperm. That's fertilization, so that's happening right here. Then we have what we call the zygote. So that's a full 46 chromosome one cell structure, which then goes through division events to become a morula on day 4 at about 16 to 32 cells. And then after about a whole week, we've got our blastocysts in the uterus implanting into the uterus to continue further development. In one of my next videos, guys, after today, we're going to be talking about the process of gastrulation, which is when the cells start to actually specialize. Because remember, at this stage so far, as a blastocyst, all those embryonic cells are identical to each other. But that's going to have to change, and the process of change is called gastrulation. Okay, we're not doing the Crayola. I haven't given you all the things to color at this stage, so we're going to quickly pass these slides that I have here. Oh, back up. Okay. Do you remember bells? They used to control your life. They don't do that anymore. But they I guess, well, no, they don't even control my life anymore, really. Guys, let's take a second to talk about the placenta. So the placenta is a structure that now needs to start its formation. And its formation is going to come out of those trophoblast cells that we talked about surrounding the embryo. It is going to take about 3 months to fully form, and it's going to be the baby's lifeline to the mother. The mother's going to provide nutrients through the blood, through the mother's blood, to the baby's blood, and that's all going to happen at the placenta, and then it's also going to help remove the waste that the baby creates. Okay, so I've got about 3 minutes left in this little video to see if I can talk to you about these extra embryonic membranes that are going to be forming as the embryo grows and develops. Part of your job, guys, is to be able to take a diagram like this or an artist's rendering and know what the structures are and what they do. So we're going to be talking about the chorion. So guys, chorion is going to be and it's going to help when we have a picture, the outer layer of cells of the growing embryo. And the outer layer of cells, and these are going to mesh with the mother cells of the endometrium to eventually form the placenta. So that's its most important job, as well as it creates the human chorionic gonadotropin hormone that helps tell the mother's brain that, hey, we have a baby developing here. We want to keep our corpus luteum, and we want to keep the flow of progesterone so that we don't lose this baby, we don't lose pregnancy, and we don't lose the endometrium lining. So there's that note there about the human chorionic gonadotropin. All right, now there's a sac that surrounds the embryo as it grows and develops, and that's called the amnion. It's an inner lining of cells. It fills with a fluid to help the baby float, protects it, controls the temperature, supports it. And you've probably heard of the water. A woman, when she's about to give birth, her water breaks. Well, that's the amniotic fluid that is found inside that sac. Okay, so we also have what's called the yolk sac, which is thought to provide red blood cells initially before we have a fully functional heart, and blood cells coming from bone marrow. Okay, there's the embryo itself. There's the amniotic sac filled with fluid, and over here will be the chorion. Chorion produces what we call these projections called villi or fingers that actually are able to mesh with the mother cells. Okay, so I talked about that yolk sac. The allantois. Haven't mentioned the allantois yet, but that's the allantois. It's the beginnings of the umbilical cord, and I'll show you that in a minute. And here we go. So guys, we're going to kind of end by looking at this diagram really quickly. There we have the embryo. This is the allantois, which will become the umbilical cord, the yolk sac, the chorionic fingers that become the placenta. There's the amnion, inner layer, fills with fluid surrounding the baby. And there we go. I have to end up my time right now so I can upload this video to YouTube. Thanks for listening, guys, and we'll see you in the next episode.